Okay, so thank you, Don, for that very kind introduction, and thank you to all of you for coming today. I think it's equally nerve-wracking and exciting to see so many faces that are important to me in the audience today. So I doubt it will come as much surprise to you that our landscapes are changing. You can see satellite images of some examples of some of those changes here. And on the broadest level, I'm interested in how plants, being relatively sessile organisms, respond and adapt to changes like these in their local environments. And we know that plants have been doing this for a very long time, and that large environmental perturbations are not new. So for example, here in Wisconsin, where we're going to focus today, we know that much of the landscape was glaciated and then recolonized by flora and fauna. But there are two major differences for organisms responding to this type of change and these types of changes. The first is that most of these before and after photos here span a few days or a few centuries at most. So these changes are happening relatively rapidly and often with greater intensity and greater frequency than organisms are currently used to. The second thing is that post-glaciation and pre-European settlement, most ecological communities occurred in relatively large and often contiguous patches, whereas today, everything that's purple on this map here is either agricultural or urban. And the complex relationships between gene flow and natural selection make it difficult to predict the evolutionary consequences of such extreme habitat fragmentation like this, and therefore how these populations might respond to changes in their future environments. So I'm going to introduce you now to this interplay between gene flow and natural selection. And this really is at the basis of a lot of my dissertation work. So when environments are generally stable, natural selection acts to locally adapt populations to their unique environments. Hypothetically, or in a cartoon format, that might look something like this, where the plants with the blue traits or the blue alleles have high fitness in the cool environments, where they perform well there, and then have lower fitness in the warm environments. Another way to think about this is that the mean phenotype in each population is also the optimal phenotype, and this is true for each unique environment. So we see widespread evidence of this in local plant or in natural plant populations. Local plant populations perform better than away plant populations 71% of the time in a large meta-analysis. So in these cases, gene flow can actually limit local adaptation by introducing maladaptive alleles and by homogenizing the regional gene pool. However, when selective pressures shift, in this case we're going to think about shifting the climate gradient, as we know is happening today, mismatches are often created between the phenotypes and the local environments. So in our cartoon, that might look something like this. And in these cases, gene flow can actually help to readapt populations by moving alleles across the landscape. So for example, from other areas of the species range. Gene flow can also help to boost genetic diversity and limit some of the deleterious fitness consequences associated with inbreeding when populations become small and isolated in fragmented landscapes. And I'll just note quickly that when managers, so these are natural processes, but when managers are purposely moving alleles across the landscape, like in that middle photo, we often refer to that as assisted migration. Some of you might have heard this term before. And then when managers are purposely introducing genetic material or genetic diversity to a population, we often refer to that as genetic rescue. And then it's important for you to know that if selective pressures or migration rates are too low and population sizes are small, genetic drift will overwhelm the force of natural selection. And so in this case, the alleles that are prevalent in a population are not prevalent because they confer some fitness advantage. They're prevalent merely because they've gotten lucky and they've won the random numbers game, and those were kind of the alleles that were randomly picked from the hat. So like John mentioned, my main goal with this work was to understand how gene flow, local adaptation, and inbreeding interact in natural populations subject to habitat fragmentation. And to do this, I used Impatiens capensis populations in floodplain forests, lowland forests, and marshes here in Wisconsin. And I'm going to give you a brief tour of some of those places right now. So many people often refer to these systems as the kidneys of our landscape. They perform many important ecological functions that both we and many other species benefit from. Some of those include reducing nutrient and sediment loading into waterways. They also promote regional species diversity. So they often have much higher species diversity than we would expect given their size and compared to other community types. They often represent some of the only non-agricultural or urban areas in our landscapes. And and collectively they're estimated to provide about 25% or greater of the terrestrial ecosystem services for the surrounding areas. And then they also provide a really interesting system for thinking about genetic connectivity in fragmented landscapes, because while each patch, each of these habitats occur in relatively small patches. This is one of my study sites here, and I'm going to overlay. There's Bascom Hill, for many of you are familiar with this, it's just outside the building right here at this scale. So these are relatively small patches, but rivers still flow from one patch to the next, and may therefore maintain some form of connectivity. So with this in mind, I sampled populations hierarchically with respect to river systems. These are the 12 populations that we're going to focus on for the rest of today, and I'm going to keep these colors and shapes consistent throughout the talk, so you can start to key on those. So there are 12 populations here. They're split evenly across these 4 river systems, and those 4 river systems are nested half within the Mississippi watershed in this lighter gray, and then the other half in the darker gray in the Great Lakes watershed. And within these systems, Impatiens capensis provides a nice model. Some of the common names are jewelweed or touchweed. Many of you are probably familiar with this species. It's common to our area and native. Here we're looking at the US distribution of this plant. It also extends quite a ways into Canada up here, but that's not shown on this specific map. It's an annual, which means I could measure a plant throughout its entire lifespan, and many of the population genetic models play nice with annuals that have non-overlapping generations, as this species does. Non-clonal, meaning that when I was in the field measuring plants, I knew I was measuring a unique individual plant. Diploid, 2 sets of chromosomes. Many plants have more than 2 sets of chromosomes, so this simplified the genetic analysis some. And then it also has a mixed mating system, which allowed us to ask some interesting questions more directly about the effects of inbreeding. So for those of you that aren't familiar, the mixed mating system goes something like this, where within the same individual plant, it can produce both chasmogamous and cleistogamous flowers. The chasmogamous flowers are generally or usually outcrossed. Here you can see a bumblebee bringing pollen from presumably another plant to this flower. I say usually or generally outcrossed because that pollen could come from another flower on the same plant, but there are mechanisms in place to limit how often that occurs. And then the cleistogamous flowers don't really look like flowers, but they're these little green buds. There's an entire floral structure within that little bud, but it never actually opens and is therefore obligately stuffed. And when I'm thinking about rivers maintaining connectivity for this specific species, I'm thinking about things like large annual spring flooding events, moving seeds much greater distances than they might move otherwise. So a lot of you are probably familiar with when you touch the capsule on these, they explode. Those seeds are only flung a few meters at most. But here you can see evidence of some of the previous flooding events in this system. So these are pretty major events. The rivers themselves could also act as strips of suitable habitat, for example, along the river banks, and they could act as corridors for pollinators to move within. So first we're going to focus on gene flow, and I just want to note that it's really difficult to directly measure any one of these 3 processes. So today I'm presenting evidence for them, but not direct measurements of them. So specifically we wanted to know, to what extent are populations genetically differentiated? And then does genetic differentiation reflect isolation by geographic distance, gene flow via river networks of suitable habitat, or random genetic drift? To answer this first question, we randomly sampled 24 to 25 individuals from each of these 12 field sites. We used a temporary standardized plot that looks like this on the ground, and here's the cartoon form of that. And then we extracted and sequenced DNA and assembled loci de novo. De novo means without a reference genome, for those of you that aren't familiar. And so we're basically assembling a puzzle, or putting together a puzzle without ever knowing what the picture on the box is going to look like. So because of that, we rely really heavily on the quality of the sequence data themselves and on statistical models to assemble plausible loci. So I tested the living daylights of the parameters that we used to assemble these loci. This took 2 years of my time, but 45 years of time on the high-throughput computing network. So this really was an extensive effort, and I'm confident in the data now. And from all of that work, when all that was said and done, we obtained just under 13,000 polymorphic loci, and from each of those loci, we randomly selected one SNP. A SNP being a single nucleotide polymorphism, which is just one place in the genome where at least 2 individuals have different base pairs. And those are the genetic data that we're going to use for the rest of the talk, I'll just mention. And then we applied genetic clustering methods. So the first method that we used is called principal components analysis, or PCA. Many of you are probably familiar with this technique. It's a standard ordination technique. There's no population genetic specific model here. And the way to interpret this, what I'm going to show you in a minute, is that each individual point will represent an individual plant. They are colored again by the population that they were collected from. The shapes represent the river systems again. And points that are closer together in this plot represent individuals that are more genetically similar. So what did we find? Pretty striking population differentiation. So you can see the river or the watersheds splitting clearly from one another. So all these warmer colors are the individuals from the Mississippi watershed. On the top, we have the cooler colors. Those are the individuals from the Great Lakes watershed. Within the watersheds, you can see the rivers pretty distinctly. Here's 1, 2, 3, 4 river systems. And then even within the river systems, it appears that populations or individuals are pretty tightly clustered into the populations that we sampled them from. The next method that we used is called SNMF. This is a population genetic specific model. You can think of it kind of similar to something like ancestry DNA, where basically what the model is doing is it's assigning the proportion of each individual's genome that belongs to a predefined number of genetic clusters. So we tested the presence of 1 all the way up to 20 genetic clusters in the data set, and the best supported model is 14 unique genetic clusters. Just to orient you here, each one of these very thin bars represents an individual plant, and then there are 14 colors, each representing a different genetic cluster. So again, you can see that there appears to be relatively limited gene flow between these populations. They're pretty distinct genetically. And then finally, we calculated the classic population divergence statistic, FST. And this is going to come up a few more times, so I'm going to walk through it so that we're all on the same page. So FST ranges between 0 and 1, 0 meaning very low population genetic structure, and 1 meaning very high population genetic structure, or strong differentiation. Essentially what we're doing is we're comparing expected allele frequencies in these subpopulations to that of the pooled population. Another way to think about this is that we're interested in understanding how alleles are arranged among populations. And then there's one other statistic that I need to introduce you to that's related and also going to come up a few more times. This is FIS. So it's related to FST. It also ranges between 0 and 1. But in this case, 0 means low inbreeding, and 1 means very high inbreeding or few heterozygous genotypes. So now we're thinking about how alleles are arranged among individuals within populations. So, not surprisingly perhaps, based on the previous few slides, we found that most genetic variation occurs among populations and individuals. Our mean multilocus FST was 0.32. This is a pretty high value. Many population genetic studies find an FST of 0.1 or less, or even 0.01 or less. And then FIS spanned from 0.2 all the way up to 0.9. And this is one of the widest recorded ranges in the literature for natural populations. And then finally, if we partition this genetic variation among all of these hierarchical levels, we see that about 30% occurs among populations, and another about 60% occurs among individuals. So now we wanted to know which landscape features might explain this pattern of population genetic differentiation. And to do this, we used a model called Bayesian Estimation of Differentiation in Alleles by Spatial Structure and Local Ecology, or BEDAZZLE. And we chose this model because it has the coolest logo. No, this is actually the real logo for the model, but we chose it because it improves upon the previously widely used Mantel tests. And it's an improvement in that it better accounts for the non-independence of these paired populations comparisons that we're doing, and it also allows for non-linear relationships. So I'm not going to go into the mathematical guts of this today, but conceptually what we're doing is we're modeling the covariance in allele frequencies as a function of different ecological distances of interest and geographic distance. So conceptually you can think of the model similar to what I've written below here, where we have FST, that population divergence statistic that I just introduced you to, as the response, and then we're testing for a relationship between population divergence and, in this case, geographic distance among populations, the number of river connections among populations, and the amount of suitable habitat among populations. And then we controlled for that wide variation in overall levels of inbreeding. Just to give you a quick sense for what these ecological variables mean or where they came from, the river variable in our case was an ordinal variable, where the smallest values represented populations that were on the same branch of the same river system, and the largest value represented populations that were on either side of that Mississippi Great Lakes watershed divide. For the suitable habitat variable, we had 3 experts independently score the about 50 land cover classes in the WISP Land 2 dataset, from suitable to very unlikely that Impatiens capensis would grow there. And then we analyzed this data layer using a program called CircuitScape. And essentially what CircuitScape is doing is it's using electrical circuit theory to ask how connected are 2 populations to each other, staying within suitable habitat as much of the time as you can, and considering all possible paths between the populations. So the output of that model can be visualized like this, where we see these borders of potentially suitable habitat popping up on the landscape. And for this species, not surprisingly, they look a lot like rivers. So what did we find? Overall, that gene flow appears to be limited, at least at the scale that we tested, which was from 10 up to about 200 kilometers. We did not find a signal of isolation by geographic distance. This is somewhat surprising. Many studies find that populations that are further apart in geographic space are more genetically differentiated, but we didn't find that signal here. We also didn't find evidence that genes flow through suitable habitat, but we did find the signal of gene flow along river networks. Just to give you a quick example for what these numbers mean here to provide a little context, if we consider 2 populations that are on different branches of the same river system, that was a river distance of 1 in our case. So that has an equivalent effect on genetic differentiation as moving those 2 populations 15 kilometers further apart in geographic space. So essentially, populations that have fewer river connections among them are more genetically diverged. To test for a signal of random genetic drift, we use the simple population genetic theory that more inbred populations and smaller populations should have drifted further or faster, and therefore be more genetically diverged than the rest of the populations. And that's exactly what we find. So FIS and mean pairwise FST are positively related, and the number of private alleles and mean pairwise FST are negatively related. Private alleles are just alleles that occur in only one of those 12 populations that we sampled, and they're a proxy for population size because they're generally rare and are thus the first to be lost as populations shrink in size. And then finally, if these signals of genetic drift and genetic isolation reflect habitat fragmentation, we would expect more inbred populations to be smaller. We did not find that signal, but I think it's because population size is a difficult thing to measure accurately, even when you have great field assistants crashing through the underbrush with GPS units for multiple hours. But we did find the expected relationship where more inbred populations occurred in landscapes with generally less suitable habitat around them, and where that habitat was more fragmented. So we found strong population differentiation, limited gene flow along rivers, more inbred populations in smaller, more fragmented patches, and collectively these results seem to suggest that genetic drift is playing a strong role in structuring these populations. So now we're going to move to thinking about local adaptation. Remember, here's our cartoon of local adaptation, and that too much gene flow can limit local adaptation. So that doesn't seem to be what's happening in these populations. Instead, we found evidence of genetic isolation. So on the one hand, genetic isolation can promote local adaptation. But remember that if genetic isolation is too extreme, genetic drift can take over and overwhelm the force of natural selection. And remember, in that case, it's just we're kind of randomly pulling alleles from a hat. So we have a rich literature on the molecular genetic consequences of habitat fragmentation. And we also know quite a bit about patterns of local adaptation in natural plant populations. But there are relatively few studies that have looked for signatures of local adaptation where the population genetic consequences of habitat fragmentation are known. And this makes it difficult to predict at what level of genetic isolation populations might become pushed over this loss of adaptation edge. So with this in mind, we asked to what extent do individuals from different populations vary in morphology and or fitness in wild populations and when grown in a common garden. So we're shifting now from thinking about molecular genetic differentiation to morphological differentiation. And then we wanted to know, is there evidence that morphological differentiation reflects adaptation to local conditions? Adaptive morphological variation can arise as a result of phenotypic plasticity, and it can be genetically encoded. And these have different evolutionary implications for the trajectory of the population. So plastic responses are generally responsive, They happen relatively quickly within one lifetime, but they're often difficult to pass on to the next generation. On the other hand, genetically encoded responses are generally fixed. So for example, we can't change our eye color. Changes in these traits happen over a longer time period, over many or multiple generations within a population, but they can often be inherited or passed on to the next generation. And one of the main tools that we use to tease apart plastic responses from those that are genetically encoded is a common garden. The main idea with a common garden, we're generally working within, or often working within one species at a time, is that we sample individuals from that species from a bunch of different environments, and we plant them all in one common environment or common garden. And then there are two main possible outcomes here. One, that individuals still look different, suggesting that those traits are genetically encoded. Or two, that all the individuals look relatively similar now, suggesting those differences in the field were the result of plastic responses to those with different environmental conditions. So this is exactly what we did. We measured morphological traits and reproductive output from 96 plants now in each of these standardized plots in the 12 field sites. And then we collected seeds from those plants and brought them back to Madison. And a quick shout out to my mom here for emergency sowing thousands of these little seed bags and mailing them overnight so that we could collect enough seeds because they explode, and if you're not there when they explode, then you don't have the seeds. So thank you to her for that. This chapter probably would have failed without those. And I just want to note that from this point forward, we tracked each one of these seeds individually throughout the duration of their lifetime in the experiment, because why wouldn't that be a good idea? So we planted all of those seeds in the UW-Madison greenhouse and germinated them there. We measured a variety of seedling size traits and whether or not the seedlings had disease or not. And then we transplanted all of the living seedlings out to the common garden, which was in the UW Arboretum. About every 3 weeks throughout the field season then, we came back and collected these data so that we could obtain for each individual plant the maximum height that it achieved before it died, the maximum stem diameter, some leaf size and shape traits, about when each plant died, the total lifetime number of both mature CH and CL seed capsules that each plant was making, and then we scored throughout the season whether or not they had any disease or galls. For this section, we're focusing only on the Toczydlowski progeny, so remember those are the obligately self-progeny, and we're doing that because we don't want to be analyzing alleles that are coming in from outside the populations for these analyses. And then we fit this mixed model to ask, do populations show differences in traits and or fitness in the common garden? And they did. So here on the left, we're looking at the percent of the total seeds collected in each population that germinated. You can see there's a pretty wide range there. And then on the right, where this is reset now just for plants that made it into the common garden, and this is the percent that were alive in each population throughout these different time points in the field season. So again, you can see that plants are dying at generally different rates depending on the population that they were collected from. Populations also show morphological differences in the common garden and differences in that total amount of reproductive output. So each one of these black points here represents a least squared mean. They're bracketed by a 95% confidence interval. So if those confidence intervals are non-overlapping, the populations are significantly different. And I just selected a few of the total traits we tested here. So moving from kind of early to late life stages, we're looking at from top down seed mass, seedling height, specific leaf area, final height or total maximum height, and then that total number of seed capsules produced. And while populations showed significant differences in the common garden, they also converged considerably from the field sites. So the black points here are the exact same on the left and the right. All I've done is add on in colors the mean values for those mother plants in their natural field populations. So populations show divergence in traits in both the common garden and the field sites. Now we wanted to know, is there evidence that these differences are adaptive? And we asked this a few different ways. The first is that we just fit this model to test, is there a relationship between traits in the common garden, the trait values of each plant, and the amount of seeds that they're making, or the number of seed capsules that they're making? The answer is yes. Traits related to fitness. Taller plants with thicker stems and bigger leaves made more seeds. And interestingly, the relationship between specific leaf area and total reproductive output varied depending on the population that the individual was from. And then not only did traits relate to fitness, but they related to fitness in biologically expected ways. So I'm just going to show you one example of what I mean by this. Plants had smaller, thicker leaves in drier and brighter environments. So this is the biologically expected response, and this is true for both progeny in the common garden and mothers in those wild field populations. And then finally, if trait differences reflect adaptation to local conditions, we would expect plants growing in environments, originally growing in environments very different from the arboretum, that's this y or x-axis here. We would expect those progeny to look very different than their mothers did morphologically in the field sites, and that's the y-axis. And we do find this expected positive relationship. However, plants that were essentially growing far from home environmentally still produced fewer seed capsules in the common garden. So this seems to suggest that there are limits and or costs to plasticity. So we found that populations differed in morphology and reproductive output in both the field and the common garden. There was evidence that these morphological differences are adaptive, and collectively populations showed patterns consistent with both adaptive plasticity and local adaptation, despite strong genetic drift. So it doesn't seem like genetic isolation is extreme enough yet where drift has totally overwhelmed the force of natural selection and erased these signatures of local adaptation. So now we're going to move to the final section on inbreeding. So we were just thinking about how if populations become small and genetically isolated, genetic drift can overwhelm the force of natural selection. The other big concern with small, genetically isolated populations is inbreeding. There are two main theories proposed for how individual inbreeding limits fitness. One is that inbreeding is exposing recessive deleterious alleles. The other is that it's limiting the frequency of inherently more fit heterozygous genotypes. And I should just note that inbreeding in general you can think of as individuals becoming more homozygous. So there's a lot more empirical evidence for directional dominance, so that's what we're going to focus on today. So inbreeding is exposing recessive deleterious alleles. We refer to those as the genetic load, and that's causing a decline in fitness, which we refer to as inbreeding depression. So we have a pretty good understanding of how this works, even in natural populations, but we know a lot less about what happens to the genetic load after it is expressed. And there are kind of two forks in the road here, potential forks in the road. So it could become purged and or it could become fixed. And what I mean by the genetic load becoming purged is I'm thinking about natural selection weeding out or purging these recessive deleterious alleles from the population. But while this is happening, deleterious alleles could also be becoming fixed via background selection. So the idea here is that it might not be we have the choice between a good allele and a bad allele. It might be that we have the choice between a really bad allele and a kind of bad allele. And so, in weeding out this really bad allele, we've now fixed all of the remaining individuals in the population for the kind of bad allele. And if the strength of selection against these mutations is less than 1/2 the affected population size, genetic drift will take over and randomly fix about half of those and randomly purge about half. And one of the reasons that our understanding of these processes is limited is that all of these have the same signature in that they all reduced inbreeding depression. So I'm going to show you quickly mathematically what this looks like. This is classic inbreeding theory where on the y-axis we have fitness and on the x-axis we have the individual level of inbreeding, and we expect this linear decline in fitness as individuals become more inbred. We refer to that decline again as inbreeding depression, and we often measure it by comparing the relative fitness of self to now-crossed progeny. And then remember that purging is improving the fitness of the inbred individuals, and fixation is lowering the fitness of the outbred or outcrossed individuals. And again, note both of those lines would have the same inbreeding depression. So not only is this interesting theoretically, it also has important conservation implications for conservation management. So some people actually think that we should purposely inbreed populations to purge them of their genetic load. But if purging is not effective and or we're fixing deleterious alleles at the same time, it might be a more prudent strategy to introduce new genetic material to the population instead. So there are both theoretical and conservation implications for this work. Okay, so to better understand how inbreeding is affecting purging and fixation of the genetic load, we asked, do outcross progeny have lower fitness than self-progeny in a common garden? So basically using the mixed mating system of Impatiens to look for evidence of inbreeding depression. And then we also used a molecular marker of inbreeding to look for inbreeding depression again, but now we were asking, do less heterozygous mothers have lower reproductive output, and do their progeny have lower reproductive output? So looking for evidence of inbreeding depression across generations. And then we wanted to know, does inbreeding depression decline in more inbred populations, again as a result of purging and or fixation? So recall that both within an individual plant, it can make both CH and CL flowers, which turn into seeds that we collected and grew side by side in the common garden. So we essentially have self and outcross plants growing side by side. And from this point forward, I'm going to try and refer to the plants as self and outcrossed. Again, noting that CH seeds do not have to be outcrossed. And then I'm going to try and refer to the seeds that these plants are making in the common garden as CH and CL to hopefully limit confusion here. So we fit this mixed model, again, looking for evidence of inbreeding depression or testing for evidence of inbreeding depression by comparing these two seed types in the common garden. And we allow the potential fitness difference between these seed types to vary depending on the overall level of population inbreeding. So we did find evidence of inbreeding depression. Outcross progeny were taller and they produced more seeds than the self-progeny in the common garden. But to our surprise, we did not find inbreeding depression to decline. So it did not decline in more inbred populations, so it was not evidence that the load was being purged. And it also didn't decline in populations with more fixed SNPs, which was our proxy for how much of the genetic load might have become fixed. So we did not see evidence that the load was becoming fixed with it. So if inbreeding depression is persistent in populations, it seems like there should be pretty strong selective pressure for individuals to outcross so that they avoid these deleterious fitness consequences associated with self-ignorant inbreeding. And also to our surprise, we actually found evidence that the self-progeny were putting more energy into outcrossing than the outcrossed progeny. And this is especially surprising given it takes a lot of energy to make these large nectar-rich flowers and subsequently larger seeds. And as you may expect, and I alluded to on the previous slide, these self-progeny were generally wimpier and smaller and kind of sadder than the outcrossed progeny. So for the same number of mature CL seed capsules in the common garden, the self-progeny were making about 20% more CH seed capsules than those outcrossed progeny. Okay, and then finally we're switching, still looking for evidence of inbreeding depression, but now instead of this relatively binary metric of self and outcross from the mixed mating system, we're using individual inbreeding. And individual inbreeding is a difficult thing to measure, especially in natural populations, because it requires a deep and accurate pedigree. So instead we used average heterozygosity across those 13,000 SNP markers as an inverse proxy for individual inbreeding. So basically the idea here is that more homozygous individuals are more inbred. So we were testing for a relationship between maternal heterozygosity and her reproductive output in the field populations, and then to look across generations, we compared maternal heterozygosity, looked for a relationship between maternal heterozygosity and now the reproductive output of her progeny in the common garden. And the blue terms here just represent terms that are different from that first one. So to our surprise, we found that the relationship between maternal heterozygosity and reproductive output actually varied depending on the overall level of inbreeding in the population. So I'm going to walk you through what this means here, where we're going to start on the far left. This panel represents a hypothetical population that's relatively outcrossed, so it has a low FIS value. And in a population like this, more heterozygous mothers have higher reproductive output. So this is what we would expect. Evidence of inbreeding depression. However, if we move over here, all the way over to the right, to one of these highly self or highly inbred populations, this relationship actually flips, where now more heterozygous mothers actually have lower reproductive output. So this is somewhat puzzling. One potential explanation is that this is evidence of outbreeding depression. So it can be that when individuals outbreed or outcross, they break up important evolved complexes within the genome. Many of you might know these as epistatic effects, and so their offspring actually have lower fitness because of that. We found the same relationship in the progeny, so the top and bottom graphs here are essentially the same, but now on the y-axis, instead of maternal reproductive output in the field, we're looking at the total lifetime reproductive output of the progeny in the common garden. And now we have two lines, one for the outcrossed individuals and one for the selfed individuals. So if this negative relationship reflects outbreeding depression, we would expect, we're basically saying outcrossing gives you lower fitness. So we would expect the outcrossed individuals to have lower fitness than the selfed individuals. They clearly don't. So what Don and I think is going on is instead, and I will note quickly that there are relatively few individuals that are represented by this line, because as you might imagine, it's difficult to find outcrossed individuals in highly selfed populations. But if this relationship is true, what Don and I think is going on is that in these highly selfed populations, this measure of molecular heterozygosity is no longer a good proxy for inbreeding, but we're instead detecting deleterious mutations that are still segregating in that population. So we found that self progeny had lower reproductive output, but we're trying harder to outcross, that the relationship between maternal heterozygosity and reproductive output varied depending on the overall level of population inbreeding, and this was true both for mothers and their progeny. And collectively, that both individual and population level inbreeding influenced progeny fitness, and that populations appear to have limited capacity for purging or fixing their genetic load. So today we thought about gene flow, local adaptation, and inbreeding in natural populations of Impatiens capensis growing in a relatively fragmented landscape. And the genetic isolation and fitness consequences of inbreeding that we observed make me worried that these populations are not going to have the genetic diversity they need to respond to changes like these in time. So part of me feels that we've developed these really cool tools like next generation sequencing and hypercomputing just in time to document these populations being pushed over this loss of adaptation edge. But the other part of me recognizes that these populations are persisting despite relatively high levels of inbreeding. They've retained adaptive traits, and they can likely move within river systems. And so that part of me wants to think carefully about how we can promote or conserve genetic connectivity in fragmented landscapes, and then take a page from Darwin's playbook where he says, I can see no limit to this power, referring to natural selection, in slowly and beautifully adapting each form to the most complex relationships of life. So with that, I have many people to thank. First and foremost, to my advisor, Don. He has given me an incredible amount of latitude to develop this research program and pursue the research questions that are really of interest to me and supported me along the way, so an incredible thank you to him. To each of my committee members, Tom, Sarah, Brett, and Sean, I purposely designed this dissertation to use multiple methods, as Don was kind of alluding to in his introduction, that are often used separately, and to study multiple processes that are often considered in relatively distinct fields. So I really relied on all of their collective expertise to bring this story together. To my amazing field assistants, Astrid De La Cruz and Brad Waldorf, who many of you know as my partner, you might have sensed throughout the talk that we collected a lot of data, and it really was just the three of us collecting all of these data, and as you can tell, many of them were quite tedious and required very careful documentation so that I could tell the story that I did today. To Sarah Johnson, she spent her PhD studying these and many other floodplain forests and lowland systems in Wisconsin in the Waller Lab previously. She was an invaluable resource when it came to which Carhartts are going to protect me from stinging nettles, or how do I contact landowners. So yeah, she was an invaluable resource when it came to everything floodplain forests in Wisconsin. There are 3 graduate students from our department, Chloe Drummond, Alex McKelvey, and James McDaniel, who spent a lot of time with me thinking about how the heck do you assemble and make sense of GPS data, those are the genetic data. This was relatively new technique in the field, so the learning curve was really steep for everyone, and it was very helpful to have other brains working on these problems. I attended the lab meetings, the Scoville lab meetings, and the Gignish lab meetings, and worked with the Dina Risman's group quite a bit for multiple years. And all of these people were very welcoming to me and welcomed me into their circles and shared their knowledge and expertise with me and really rounded out my education here outside of my dissertation. So thank you to all of them. To my funding sources, the NSF Prevention's Biodiversity Grant that multiple PIs in our department secured helped a lot with the sequencing, the GRFP, The botany department has been incredibly generous in supporting me with multiple fellowships and supporting my research with multiple grants. I taught for 4 semesters in the zoology department, and they offered support there, and then the graduate school has also kicked in money when some of these fellowships haven't quite covered everything. A huge thank you to all of the members of the Waller Lab. I've been really fortunate over the past 6 years to be surrounded by some very bright, talented, and kind individuals. I'm really going to miss sharing an office with these people and trying to figure out why R won't work when I need it to, and what the heck my data means, and winning ecology trophies, brushes off shoulders. Yeah, it's really been a much more enjoyable ride because of all these people. A special and very big thank you to Steve Goldstein. He's our research computing facilitator. He spent tens, if not hundreds of hours behind the scenes with me thinking about ways to write creative code and to write code that can get a lot of work done efficiently. He's drawn pictures like this for me, I don't know how many times, to explain how high throughput computing works and to help me understand how to think in parallel, which was not initially intuitive, at least to me. He really is an invaluable resource to our department and has contributed a lot behind the scenes to this work. Another special thank you to my close friend and collaborator, Cecilia Ramon. She invited me to help her design and offer a symposium to the public that was at the intersection of art, ecology, and design. And this was a really fun and rewarding way to tangibly share this research outside of the scientific community and just my passion for natural systems in general. To all of my friends and family, thank you for all of your patience, your understanding, and your support. I would not be here today without you. A special thank you to my parents for giving me a childhood that looked like this, which is one of the main reasons that I pursued a career in biological sciences. My drive and passion for natural systems really comes from them and allowing me to be chased by otters when I was 4 years old. This is me right here in the kayak with my dad on Iron Royal. So a huge thank you to them. I recognize now that this is not what everyone's childhood looked like. And then a bigger thank you that I can put into words to my field assistant and partner, Brad. He helped me to collect all of the data for my dissertation while working a full-time job himself. And I'll just mention to you that floodplain fieldwork is a very challenging task. So everything needs to be mosquito-proof, and/or waterproof, and mud-proof, and rotten clam-proof. It needs to be metal-proof. Many of you know John Curtis. He is a famous ecologist, or was a famous ecologist that surveyed many, if not all, of the plant communities in Wisconsin. And one of the only things he writes about the non-plants in the floodplain forests is, among the lesser animals, the insects make their presence known through their ubiquitous and frequently overwhelming representative of the mosquito. So these really are challenging systems to work within, and Brad also cooked well more than 50% of the meals over the last 6 years, and was an incredibly supportive person to get me to this day. Here we are on the very final day of fieldwork. From the first year of fieldwork, collecting all of those field measurements and seeds. And then, like Don mentioned, my next adventure is a postdoc position in a few weeks in the Bradford Lab at Michigan State University, and he actually is the mind hive behind the unicorn model. So with that, I'll be happy to take any questions.